The Microbiome Service Center combines the power of our scientific expertise with an in-house metagenome application for all of your genomic analysis needs. This, combined with our metabolomic service offering, creates a multi-omics approach to answering all of your scientific questions. Let's take a look at some of the capabilities of our metagenome application pipeline. We currently support a complete metagenome analysis and are now launching a metabolomic analysis pipeline on our application as a one-stop shop for microbiome analysis. On the metagenomic side, we use a manually curated in-house database to combine alignment and k-mer classification to better characterize 16S ribosomal RNA. The novel algorithm we use generates precise species-level classifications for known 16S sequences and offers better coverage for ambiguous or unknown 16S sequences than other approaches. The app's curated database resolves ambiguities found in other publicly available databases. Once the data is uploaded, we can generate a wide variety of figures. The first data we can visualize is alpha rarefaction. This shows the observed OTUs for each read depth. As you can see, we quickly reach a sequencing depth at which all available taxonomic data is captured. The ranked stats graph shows what calls are being made for each sample at different taxonomic levels. We also generate individual sample classifications. We can visualize the classifications by cake chart and taxonomic trees, as well as in tables that can be downloaded. Each of the components of these charts can be moused over to see a readout of the classification. We also visualize the taxonomic data in abundance heat maps. The application generates stacked bar graphs as well, showing the different bacteria identified in each sample at different classification levels. A table of the taxonomic calls is also generated. This allows easy visualization of the differences between sample population, like the diseased and healthy samples represented here. We generate beta diversity graphs for the data. This shows the degree of differentiation among biological communities, sorting the samples into categories based on differences in community composition. Here we can utilize different categories in the metadata. P-values are generated for each graph. We can see that there is a statistically significant difference between the diseased and healthy samples in this study. The application also generates left-C analysis. This analysis shows the differentially expressed bacteria between two categories. We can run the left-C analysis for different categories in the metadata and generate an LDA score and p-value for each bacterium identified. This allows the researcher to identify significant bacteria species that are differentially expressed. The next step is to connect the metagenomics data to metabolomics data to get an idea of the metabolomic differences between these samples and expand on the functional significance of the data. We are in the process of launching the metabolomics portion of our application and currently generate the same wide array of figures separately. We look forward to helping you answer your scientific questions.